Hi students, today topic is amembranous cell organelles. We discuss about the amembranous cell organelles. What is amembranous cell organelle? Around the cell organelle, membrane is absent. We know about ribosome. Around the ribosome, membrane is absent. So ribosomes are amembranous structures. In the same manner, a group of organelles are there which are not having the membrane. These are called as amembranous structures. Amembranous structures, right? So in that case, there is a microtubules are there, intermediary filaments are there, microfilaments are there. Majorly within the cell, other than ribosomes. Here, microtubules, intermediary filaments, right? Microfilaments, microtubules, intermediary filaments, and microfilaments. See, these are the three structures which are not having the membrane. That's the reason why these are called as amembranous structures. And if we see in the order, right, microtubules, these are the long structures. Intermediate filaments, these are the intermediate structures. And microfilaments, right, these are the short or small structures, right. That is about the order and the size. So here this order also indicates for the size. See, these three structures especially involve in the different cell organelles formation. We'll discuss what are the types of organelles are formed by these three parts, right? So first of all, these three are combined: microtubules, intermediary filaments, right, and microfilaments. These three are combined. These are produces the cytoskeleton. See, we have to know about skeleton. Human beings are having particular shape. Due to the reason is the skeleton. Dog have particular shape because dog have a particular shape of skeleton. Buffalo, donkey, right? Different animals. See, the animals are having particular shape. Due to the reason is the skeleton, right? Here cytoskeleton. Cyto indicates here cell. Cell also having particular skeleton system. Remember, I like to ask one question here. One of the plant cell is there and one of the animal cell is there. If I ask the question, the plant cells are expanded. See, how the plant cells are expanded? Everybody will say the answer is, sir, plant cells are having cell wall. Then the cell wall giving the mechanical strength. Then the plant cells are expanded. But in animal cells, there is no cell wall. If I ask the question, how the animal cells are expanded? Animal cells are expanded or not? Definitely expanded. Every cell is definitely expanded. So here animal cells also expanded. How the animal cells are expanded? Because animal cells are majorly involved in the formation of cytoskeleton. So presence of cytoskeleton, the animal cells are expanded. See how the cytoskeleton formation takes place. I said here microtubules, intermediary filaments, and microfilaments. These three are combined, produces the cytoskeleton. Cytoskeleton is also called extracellular matrix. So how the extracellular matrix structure is formed? Simple. Microtubules are combined, and microtubules, intermediary filaments, and microfilaments, all these are combined, which produces the proteinaceous structures. See, the red color indicates here the proteinaceous structure. So overall, the red color part indicates here cytoskeleton. So presence of these proteinaceous structures or presence of these microtubules or intermediary filaments or microfilaments, see, the animal cell expansion takes place. So the expansion due to the mechanical strength. Okay, the mechanical strength provided by the cytoskeleton. Because in animal cells, cell wall absent. So the mechanical strength given by the cytoskeleton. So I said these three are supporting to form the cytoskeleton. Due to the cytoskeleton, the animal cell expansion takes place. Sir, you are talking about the animal cell only. In plant cells, cytoskeleton is present or not? Remember, always animal cells are advanced cells, and animal cells are having much complex. Animal cells are having much complex. So what I am telling here, in plant cells also cytoskeleton is present. Animal cells also cytoskeleton is present. But the animal cells contain the cytoskeleton is well developed. The animal cells contain the cytoskeleton is well developed than plant cells contain cytoskeleton. So the cytoskeleton of plant cells not well developed, poorly developed. In animal cells, the cytoskeleton is well developed because already in plant cells there is a cell wall is there, right? That is the one of the reason. So that is about the cytoskeleton. Coming to the next one is the microtubules. Microtubules, right? These are the long structures. Microtubules are the long structures which are supporting for the construction of cilia and flagella. The microtubules are involved in the construction of cilia and flagella. I will discuss. Anybody? Yeah, I thought most of the students are well known about what is the function of flagella. Flagella which are present in bacteria. Flagella which are present in the bacteria. See, especially the unicellular eukaryotes or unicellular prokaryotes. Especially these are bacteria indicates here unicellular prokaryotes. Unicellular prokaryotes which are having long hair-like structures. Okay, the long hair-like structures are supporting for the locomotion of the bacteria. Locomotion of the bacteria. See, the bacteria. See, simply I like to explain about here. Okay, prokaryotic flagella and eukaryotic flagella. Between these two, much difference is there. In prokaryotic flagella, in prokaryotes, the flagella developed by the proteins. See, in eukaryotes, the eukaryotic flagella developed by the microtubules. Especially here, the microtubules are arranged in eukaryotes 9+2 arrangement. 9+2 arrangement. For suppose this is one of the euglena. Euglena having the flagella. Euglena belongs to the eukaryotes, right? So we know about this is the structure of the euglena. Euglena having two flagellas. If we cut the flagella, the eukaryotic flagella, the cross section of eukaryotic flagella at center, 2 proteins are present. At peripheral, 9 proteins are present. At peripheral, 9 proteins are present. That's the reason why this arrangement is called as 9+2. This arrangement is called as 9+2. So in eukaryotes contains the flagella showing the 9+2 arrangement, and eukaryotes flagellas are majorly formed by the microtubules. But in prokaryotes not like that. There is a protein is there. That protein is supporting for the formation of prokaryotic flagella. That is the structural differentiation between prokaryotic flagella and the eukaryotic flagella. So here microtubules are supporting for the formation of flagella and cilia. We know about the paramecium. See, for suppose this is one of the paramecium structure. It's having that small hair-like structures or short hair-like structures. These are called as cilia. The difference between flagella and the cilia, flagellas are long structures. Here cilia are short structures. Both are supporting for the locomotion. So the construction of flagella and cilia, right, which are made up of with the microtubules. Right? That is about the microtubules involved in the flagella formation and the cilia formation. So coming to the next one is the centrioles, right? I thought most of the students are well known regarding to the centrioles. Centrioles are present in animal cells, right? Centrioles are present in animal cells, which are supporting for the spindle apparatus formation. Or suppose this is the one of the cell, right? And this is the nucleus, right? And these are the centrioles. These are the cylindrical structures which are arranged in perpendicular manner. Right? See, these are majorly supporting for the spindle apparatus formation. Means during cell division process, the centrioles are moving towards the opposite pole side. And which are produces the spindle apparatus. Like this. This is the spindle apparatus. Okay, these are the spindle apparatus. So centrioles are made up of microtubules. Spindle apparatus also made up of with the microtubules. All of you remember, centrioles are made up of with the microtubules. Spindle apparatus also made up of with the microtubules. Both are made up of with the microtubules. Then what is the function of the spindle apparatus? Okay, what is the major function of the spindle apparatus? We know that during the cell division process, the chromosomes are separated. The chromosomes are separated. How the chromosomes are separated? Or how the chromosomes are moving from cell center to opposite pole side? By the spindle apparatus. So the spindle apparatus are supporting for the separation of chromosomes. So here, spindle apparatus during cell division process involve in the separation of the chromosomes. Right? That is about the spindle apparatus. Right? Sir, first suppose in a cell, spindle apparatus are not working. What happened? Just now I said spindle apparatus are involved in the separation of the chromosomes. Then constant number of chromosomes are maintained. Constant number of chromosomes means every species having particular chromosome number. So in every species, the constant number of chromosomes are maintained by the cell division, right? So here either in mitosis, either in meiosis, right? The particular number of chromosomes are maintained. In mitosis, same number of chromosomes are maintained. In meiosis, the number of chromosomes are reduced, because especially the meiosis takes place in sexual cells for the production of male gametes and the female gametes. So here the chromosomes are separated. If the spindle apparatus are not working, what happened? Then all these chromosomes are entering to one cell. Means the number of chromosomes are doubled. In a cell, the number of chromosomes are doubled. Generally, this technique we are using for the hybrid plants production. In plants, this technique we are using for the hybrid plants production. We are eating grapes, hybrid grapes, not normal grapes, hybrid grapes. We are eating watermelon, right? We are eating carica papaya fruit, right? So these are some examples. We are producing the hybrid plants. By the increasing the number of chromosomes. Sir, naturally all cells forms the spindle apparatus. Then how we inhibit here spindle apparatus formation? By applying particular alkaloid. Name of the alkaloid is colchicine. Colchicine is one of the alkaloid. That alkaloid we are extracting from colchicum alternate. Right? This is one of the plant. The plant contains rhizome. From that rhizome, we are extracting the colchicine alkaloid. That alkaloid applying to the particular plant cells, the spindle apparatus are inhibited. Then the number of chromosomes are doubled. In a cell, the number of chromosomes are doubled. Then that particular cells we are using for the hybrid plants production. That particular cells we are using for the hybrid plants production. Right? That is about that spindle apparatus formation inhibition. Generally, here microtubule size is the 25 nanometers. The microtubule size is the 25 nanometers. All of you remember, microtubules are very important structures which are supporting for the formation of cilia and the flagella. Both these are belongs to the eukaryotes, eukaryotic flagella, right? And cilia are seen in only eukaryotes, but flagella is also seen in prokaryotes. But prokaryotic flagella is different from eukaryotic flagella, right? And centrioles, these are seen in animal cells. Centrioles are majorly involved in the spindle apparatus formation. Centrioles are made up of with microtubules. Here, spindle apparatus also made up of with the microtubules. These are also called tubulin proteins. Right? The size of microtubules is 25 nanometers. Right? That is about the microtubules. And next one, intermediate filaments. And next one is the intermediate filaments. Yeah, intermediate filament means the size is intermediate. Here the size is the 8 to 12 nanometer size. These are present, okay, these are especially, okay, these are the examples for the intermediate filaments. Keratin filaments. Keratin filament means our skin having the elastic nature, because the skin having elastin proteins, along with that keratin filaments also present. The skin majorly composed with the elastic proteins, right, and keratin proteins. Keratin proteins also seen in nails. Keratin proteins also seen in hair. So hair, nails and skin majorly made up of with the keratin filaments. Here examples are skin, right, nails, hair. These are majorly made up of with the keratin filaments. Neurofilaments means the filaments which are present in the neuron. We know about that neuron structure. There is a myelin sheath is there. The myelin sheath is majorly made up of with the neurofilaments. The myelin sheath is majorly made up of with the neurofilaments. And coming to the localization of cell organelles, this is a very important function. See, in a cell, different cell organelles are there. Almost all cell organelles are fixed to one particular position. All cell organelles are fixed to one position. Sir, how cell organelles are fixed to one position? How cell organelles are fixed to one position? That fixing position done by the intermediate filaments. Especially the intermediate filaments are supporting for the localization of cell organelles. Right? That is about the intermediate filaments. And last one is the microfilaments. The size is too short, 6 to 7 nanometers. Right? Actin filaments and the myosin filaments. Actin filaments and the myosin filaments. So where we have seen the actin and myosin filaments, especially skeletal muscle. Skeletal muscle majorly made up of with the actin and myosin filaments. See, the actin and myosin filaments. First, suppose these are the actin filaments, these are the myosin filaments, right? Within that skeletal muscle cell, the sliding takes place. This is the contract state. This is the relaxed state. Or suppose this is the hand, this is the contract state. So this is the muscle cell contract state, skeletal muscle cell contract state. This is the relaxed state. So this is the relaxed state. So the muscle contraction and the relaxation done by the sliding of actin and myosin filaments. So the actin and myosin filaments belongs to the microfilaments. Actin and myosin filaments belongs to the microfilaments. Right? That is about the membranous cell organelles. Right? Next class we will discuss about the ciliary structure, flagella structure. Right?